Можно начинать, да? Так, уважаемые коллеги.
Dear colleagues, we're beginning the panel session for the defense of the thesis by Anton Bankevich. The thesis is submitted in conformity with the requirements for the degree of the candidate of science. The specialization is 050318, mathematical modeling, numerical methods and complexes of programs. Under the order issued by St. Petersburg State University, number 2761/1, I, Sergei Andrianov, have been appointed the chairperson of the Dissertation Board. Let me introduce my colleagues. Professor Elena Kotina, Doctor of Physical and Mathematical Science, Professor, Department of the Control Theory, and Andrei Mironov, Doctor of Biological Science, Moscow State University. Dmitry Chudakov and Alexander Degtyarev are attending the session remotely via Skype. So, under the federal law as of the 23rd of August 1996, St. Petersburg State University is entitled to confer academic degrees, but we have started doing it only recently. The corresponding order 6821/1 was issued on the 1st of September 2016. According to the above-mentioned order, the session of the Dissertation Board is being held. So the session is valid, providing 2/3 of the appointed members of the board are present. The Dissertation Board consists of 5 people, 5 of which are present, including 2 people who are attending the defense remotely via Skype. Therefore, we have a quorum. The agenda is as follows. The session should not exceed 2 hours. Number 1, the chairman's presentation about the documents submitted by the candidate for the degree. The candidate's presentation, 15 minutes and not more. Question to the candidate regarding the presentation. Report on the thesis. All the reports are posted on the website, so I ask you to read out only the critical remarks. Professor Kotina, Professor Mironov, Professor Chudakov, and Professor Degtyarev, I will be the last person to speak. The candidate's comments about the report on the thesis, open discussion about the presentation, no more than 5 minutes for each speaker. So it is necessary to fill in the registration form and give your full name before the talk. The candidate replies to the questions, no more than 5 minutes. The next item on the agenda is the presentation of the candidate's thesis supervisor. The candidate supervisor is not attending the session, so I will be reading out the report. Then a 5-minute break before the open balloting. During the break, the session is not broadcast. Now, open balloting, vote counting, and the recording of the results in the minutes of the session. Making a decision on whether to confer or not to confer the academic degree, and the candidate's closing speech. We'll have a short technical break.
А я так думаю, сейчас разгонимся быстренько. Все нормально, да? Итак, приступаю к ведению заседания.
So let's proceed with our agenda. I'm going to read out the list of documents. The thesis is submitted in conformity with the requirements for the academic degree of the candidate of science. The topic is mathematical models, algorithms, and software for genome assembly from complex sequencing data. The thesis was approved for the defense by the relevant order of the Academic Secretary of St. Petersburg State University. The dissertation board is set up in accordance with the order issued by the Academic Secretary, number 2768/1, issued on the 10th of April 2018. I would like to introduce you my colleagues, Professor Kotina, Professor Mironov, our colleague from the Czech Republic. Please introduce yourself. This is just to check the microphones. I'm Dmitry Chudakov, and my other colleague, I can hear you, but I cannot see you. I can see you as well. So the candidate has submitted the following documents: an application in the name of the rector, Professor Nikolai Kropachev, with a request to examine the thesis and accept it for the defense. Report of the supervisor, candidate of physical and mathematical science, Professor Pavel Bevziner, list of published works which describe the major research findings. 6 academic papers have been published in peer-reviewed journals recommended by the Minister of Science and Education. Letter of verification certifying that Bankevich Anton was enrolled in a postgraduate degree course. The specialization is 050318, mathematical modeling, numerical methods, and complex of programs, and passed 3 qualifying exams with excellent marks. Thesis in Russian and English, a printed version with a manuscript copyright, and a digital copy. The thesis formatting and reference list comply with the state standard, and also a copy of the higher education certificate. All the above-mentioned documents comply with item 12 of section 3 of the order on the procedure of granting academic degrees at St. Petersburg State University. Now, I would like to give the floor to the candidate for the degree. You have 15 minutes. And questions only about the candidate's presentation.
We do not have any external reports. You're welcome. Микрофон. Нажать кнопочку внизу. Good afternoon, my name is Anton Bankevich, and today I'll tell you about the major research findings. First off, I'll provide general outline, the problems I had to solve, and I will describe major approaches of mathematical modeling which are used in this field. Then I'll provide a brief description, the 2 basic problems I focus on in my study. This will be followed by the description of software which allowed to implement the methods and algorithms I developed. I focused on the DNA molecules. DNA is a molecule which is a major storage of genetic information of human organisms. A DNA is a sequence of nucleotides. There are 4 types of them: adenine, guanine, cytosine, and thymine. DNA may be encoded as a sequence, as a string above the alphabet of 4 letters. Unfortunately, cutting-edge biochemical technologies do not allow to completely reconstruct a DNA string. However, there are technologies which allow to reconstruct short substrings of a larger string. This is where a problem arises: how to reconstruct a complete genome string from shorter substrings. This is a computational problem. Different mathematical models are used to solve this problem. The most popular ones is a graph model. In a graph, every edge is a string, and genome is a path in this graph. And the genome graph is reconstructed as a combination of strings as edges in this graph. Another model is a probabilistic model, where every read is represented as the multiple reads as a set of random variables. I will start with the first problem. This was the problem of false edge detection in de Bruyn's graph. A de Bruyn graph is a directed graph. It's used in genome sequencing and mathematical modeling of genome sequencing. The vertices of the graph correspond to strings of the fixed length k, called k-mares. Directed edges of the graph correspond to strings of fixed length k plus 1 mares. K plus 1 mare, x connects prefix and suffix of x of length k. Graphs are usually compressed. All non-branching paths are compressed into one edge. So thus the edge gets its length, that is the number of k-mares which shape it. A genome path is a path that is shaped by a sequence of k-mares in a genome string. The multiplicity of an edge is the number of times the genome has passed through this path, and repeats. Those paths have vertices with outgoing and incoming degree of 1. Some false edges can arise. They do not pass through genome path, and their multiplicity equals 1. The number of these false edges is much higher than the quantity of real edges. Thus, we face the problem of detecting the false edges, and it's really crucial for genome assembly. There is a standard way to solve this task. This is an approach based on the coverage of edges. Edge coverage is the number of reads that is supported by this edge. For standard sequencing data, it's pretty easy to detect erroneous edges based on the coverage data. We can suppose that the distribution of coverage of correct edges follows the Poisson distribution, whereas erroneous edges follow the distribution concentrated at 0. The particular type of the distribution differs depending on the problem. So false edge detection is an attempt to estimate the parameters and to find a threshold that will allow to separate erroneous edges from the correct edges. This approach doesn't work on non-standard data. when we deal with single-cell sequencing, this model needs modification. There, the coverage is almost the same. So in my study, I suppose that we should work with the length of edges, but not the coverage. False edges almost always have a short length, no longer than 150 nucleotides. Very long edges often have the multiplicity which equals 1, and they seldom repeat. This supposition does not allow to detect erroneous edges directly. However, here additional computational methods can help. The theory of graphs has to be supported by additional computational methods. We can develop a system of linear relations which comprises the condition of balance. So the total multiplicity of outcoming edges equals the total multiplicity of ingoing edges. And secondly, lower and higher bounds of multiplicity have to be introduced. Thus, multiplicity of edges can be represented as the circulation in the Brun graph. So the solution here lies in the analysis of circulation, and we can state that all the edges which have zero multiplicity in any circulation with the bounds I have specified, these edges are deemed erroneous because they have zero flow in any circulation. So I offer an algorithm which allows to detect false edges. I have also introduced additional heuristic methods which allow to effectively apply this algorithm on real data. I also provide a theoretical estimation of algorithm efficiency. It shows that the algorithm has high specificity in normal conditions. I also provide the results of numerical experiments which show that in reality the specificity is higher than theoretical estimations. Major results of this part of my study are given in Chapter 2. This is the mathematical model of single-cell sequences based on the Brun graph, heuristic algorithm of false edge detections, the estimation of the effectiveness of the algorithms, and theoretical estimation and the results of numerical experiments, and the software were complex to implement the algorithms. In my second problem was about the metagenome size estimation in multi-bacteria community. It's difficult to differentiate between one species of bacteria and another species. In this case, their genomes are sequenced all together. It's called metagenome sequences. Some bacteria are represented in a great number of reads if they were highly represented in the sample. Some bacteria can have low representation, and we only see a few reads, which makes it difficult to assemble their genome. So the challenge was to reconstruct as much information as possible. And the problem we focused was as follows: to find the total length of the genome of the bacteria, that is to obtain the metagenome, the bacterial metagenome. Here we used two technologies, Illumina and TSLR technologies. Illumina reads are usually from 150 to 250 nucleotides, whereas TSLR technology reads are from 1,500 to 12,000 nucleotides of reads. For each of the technologies, sequencing data are represented as a sample of random variable. The value is the position in some of the genome of the bacterial community. There is a certain density of this distribution, and our assumption was that the density of distribution is changing slowly along the genome, and we also applied the aligned function. This allowed to find correspondence between the TSLR and Illumina reads retrieved from the same area of a genome. We observe only the values of the aligned function on all TSLR reads. We designed formulas to obtain the size, the total genome, PL, where L is a TSLR read, P is the density of a random variable C, and we also developed a formula for the PL value. We also introduced the method of approximate estimation of these values through the observed data, and this is the value of the align function. These two approximate formulas can be used together to evaluate the size of the metagenome. The calculated estimation was proved consistent, however, it was biased. So we analyzed the bias value and revealed if the coverage is enough, the relative bias does not exceed 10%. We applied these methods on real data, and we obtained preliminary numerical estimates of the metagenome of a human intestinal tract and the size of the metagenome of soil. The metagenome of soil has already been in focus of scholars for some time, and we believe that the estimates we obtained are more accurate because of the method we used. So the major outcomes of this part of the study are provided in Chapter 3. This is approximate calculation of the total bacterial genome length in the metagenome, the calculation of the bias value of the estimation, numerical experiments to evaluate the effectiveness of the estimate, and the software. Let me say a few words about the SPADE software. It was written in Python and C++ languages. Our task was to develop a universal tool for genome assembly, the one that could be adapted for different data types. So to solve this problem, we developed the software. It includes several modules. It's really effective and doesn't require that much computational power. On this slide, I provide an approximate step-by-step process implemented by SPADE. I was not the only person who developed this software. It's an outcome of the efforts of a team of scholars from St. Petersburg State University, and my contribution here was the participation in the design of the structure of this software, and I also was responsible for the algorithms which are described in the dissertation. The results of my study were tested. The results and research findings were discussed at international conferences. They were published in Scopus and Web of Science indexed journals. These scholarly works are cited, and SPADES is in demand worldwide. Thank you for your attention, and I would like to thank my colleagues from the laboratory. Now we are taking the next item on our agenda, and we are focusing on the reports on the thesis.
Now, please focus on the critical remarks. Thank you. First of all, I would like to say that the thesis is relevant, since modern biological and medical research also require genome assembly. I also find it important that the algorithms have been implemented within the software complex, which is used in many laboratories and institutions. And this is proved by the relevant publications. Among the critical remarks, I would like to focus on the following. The algorithms are not given in detail, and I think you need a deeper mathematical analysis of the objects of study. And there are also some stylistic inaccuracies and misprints. However, I believe that the thesis of Anton Bankevich, Mathematical Models, Algorithms and Software for Genome Assembly from Complex Sequencing Data, complies with the major requirements set forth in Order on Granting Academic Degrees at St. Petersburg State University, and Anton Bankevich can be awarded the academic degree of the Physical and Mathematical Sciences. The specialization is 050318, Mathematical Modeling, Numerical Methods, and Complexes of Programs. Now please give your reply to the comment. Thank you very much for your comment. Yes, I agree with your critical remark. I believe that a complete analysis in such a complex thesis is a challenging task. But I've done everything possible. I've done everything I could. I have a question. The genome assembly is a difficult task which was first undertaken in the 80s. And in those times there were few reads, and we did not study genomes, we studied separate genes. With the increase and with the tasks becoming more complex, our task is very relevant. I mean the task of genome assembly. And the development of the technology, and the thesis is devoted to the development of technology. is quite relevant. Today there are different methods of genome assembly, and new genome assembly technology require new approaches, and the thesis describes this new approach. Now let's move on to the critical remarks. I will start with the following remark. It's about the overall length of the metagenome. Maybe this task is not quite correct, but I have a feeling that we cannot estimate the full length of the genome. There are some impurities, but this is a different issue. However, the thesis gives an approach for the approximate estimation of the length of the genome. Now my critical remark. So the introduction speaks about some early mathematical models. I think this information is superfluous. However, some methods, such as methods by Maxim Gilbert and Sanger, have not received a full coverage in the thesis. My next critical remark, page 13. You write, Illumina technology produces random substitutions with the probability to change a nucleotide. Well, I think this is not quite correct. We think that this line has some inaccuracies. Page 15. Contexts do not overlap. This is a quotation. I don't think that this is correct, because you need a more accurate wording here. There is a small remark. You write about strains, and you should have used a more accurate term here. And there's also a phrase which seems quite vague. You haven't just used a term, and you write that this length does not exceed 95% and does not exceed 90%. And it is only now that I have understood this phrase, so you should have reworded it. But in general, the thesis is well written. I believe that the computational methods are quite correct. We understand that biological objects are quite difficult to analyze by computational methods. So I don't quite agree with the previous speaker. I would like to say that we have a complete product here, and this is not always the case, and this is not a common thing. It is not common for mathematical thesis to describe a finished product. So I believe that the thesis of Anton Bankevich complies with the major requirements set forth in the relevant order on the procedure of granting academic degrees at St. Petersburg State University, and Anton Bankevich can be awarded the academic degree of the candidate of science. The specialization is 050318. That's all for now. You may reply. Thank you for your commentaries. I understand your criticism of the introduction, but I try to show the link between the earlier models and modern models. I understand it looks quite strange. As for your question about the probability of errors in the nucleotides, yes, the probability falls at the end and it increases at the beginning, but we did not base our thesis on this assumption, and there are fewer errors in the read and there are more errors at the end of the read, but it is rather positive side than a negative side. As for the contigs that do not overlap, I would like to say that it is difficult to formulate the task that we have set up. And when you try to use computational methods to describe the task, we have to make some assumptions that probably do not have much influence on the finished product in terms of biology. And as for the phrase that you did not understand, there was a misprint. This is part of a genome of a bacteria which corresponds to the repeated sections. It does not exceed 10% and not 90% as is written in the thesis. So that's all I wanted to say. I'm happy with your replies. Now the floor is given to Professor Chudakov.
Can you hear me all right? Is it any better? I'll do some shouting then. I'm not going to read out the complete report. It's pretty long. I have numerous small critical remarks. I really enjoyed reading this thesis. It's a part of something bigger than Anton Bankevich's contribution. It's part of a teamwork. So his contribution is really huge. I have a general positive impression of the study. Getting back to the comment of the previous speaker, it remains unclear to me why the quality of sequencing is not taken into account. There is nothing said about that in the dissertation. Today, I haven't heard the reply of the candidate. This information is subject to change during reads. It doesn't mean whether we use Illumina technology or any other technology. There is always a chance, a probability of an error, and we have to account for this probability. Modern sequencing technologies more and more often use barcoding, OSR technology, for example. Barcoding used for every fragment, the so-called molecular barcoding. It allows to increase the quality of sequencing dramatically. That is to decrease the number of errors. When there is no molecular barcoding, if we're talking about genome sequencing, and there is some amplification taking place, then there is a number of reads which are characterized by one and the same point of the first read, which allows to uniquely identify starting fragment. So sequencing reads that belong to one and the same starting read in many approaches to sequencing allowed to group reads and to dramatically increase the quality, that is to decrease the great number of errors before the major analysis is performed. This is something that the candidate has not done, and the sequencing quality in the developed algorithms is not considered, which I personally find odd, and this at least requires a comment on behalf of the candidate. This is one major question that I have. I suppose I'm a biologist, and this study is purely mathematical in its character. In many cases, I understand that there was not enough analysis of how this or that protocol works on a molecular level. This allows to understand how to correct errors and how to work with this data. It is important that bioinformatics goes hand in hand with those people who prepare these libraries, so that each party understands the importance of this event. One of the examples it is given in the thesis that chimeric reads and edges sometimes can be found in standard sequencing libraries. Produced without application of amplification. As far as I know, in genome sequencing, they prefer using amplification technologies. So chimeras can be found not only in single-cell sequencing. They can be found in any genome sequencing data. Even if you use amplification-free technology, still you can find chimeric edges because they appear on the stage of when adapters appear. This is one of the examples. Chimeras appear during the ligating so this was something that I didn't quite understand. The same refers to figure 2.1. This describes some filters must be used in the consecutive analysis, but when I analyze this figure, I see some inaccuracies. So the question is in general about the attachment of next-era adapters. I will stop with my criticism at this point. In general, the dissertation is written in conformity with all the requirements, and Anton Bankevich can be granted the academic degree of the candidate of the physical and mathematical science.
I would like to answer your question. So your first question was about the quality of the nucleotides, which give traditional information. The point is, this space software includes several algorithms, and the information about the quality of the nucleotides is used at a preliminary stage. There's a tool which is called base hammer, developed by our laboratory. This tool allows us to correct the reads at a preliminary stage. Can I interrupt you? This tool that you are describing unites the reads that have the identical starts. Is it correct? It maximizes the quality. It is not quite correct. The tool doesn't take the reads that have one start. It looks at the chimeras and then it takes the substrings that came from the same position in the genome, compares the substrings and chooses the correct and the erroneous ones. This is what you are talking about, but it is at the level of chimeras, not at the level of reads. I did not participate in the development of this tool, so that's why I did not include it into the thesis, but I gave only a passing remark about it. So as far as I understand, the quality is improved before your analysis. Yes, that's right. The second question was about the biological analysis at the molecular level. I agree that this is a very important issue to understand and to develop effective methods of error correction. Unfortunately, our lab and myself deal with computational methods and algorithms, and we cannot study it at a molecular level. To this end, we would have to cooperate more with biologists, and I understand that we lack this cooperation with biologists. But everything that can be done in terms of algorithms has been done in the thesis, but you have given a very interesting task, which is not given in the thesis. Thank you. Are you satisfied with the replies? Yes, I am. Now the floor is given to Professor Degtyaryov. Good afternoon, my colleagues. I'm the last here to speak, and it's difficult and at the same time easy. Can you hear me? So what I want to say, first of all, it was difficult for me to understand every detail in the thesis because I'm not an expert in molecular biology. I'm an expert in mathematical modeling. When I looked at the thesis, I realized that the thesis did not deal with the specialization 0503-018. It is a difficult task, and it was a difficult area for me as well. What I can say about the thesis, so I try to assess it formally from the point of view of its methods and correspondence to the specialization. Unfortunately, I did not see a great number of models. However, after talking to the candidate for the degree and after hearing to the reports, I can say that the thesis is quite valuable and that you do not need so many models there. For example, in the second part, you divide between the overlap areas and different other areas. There are different methods which are in one frequency zone, and as well as other methods which have been developing recently. But here you have built a very simple model which doesn't contribute to the mathematical modeling. Maybe from the point of view of biological information, it is a new approach. But at the same time, the thesis is quite relevant and important. And I would like to say that the complexes of software that are presented here are not given in much detail. For example, they do not give information about the storage or about their cooperation with each other. For example, you don't speak about parallel algorithms and different other things. So I cannot say that I see any new or specific results which correspond to the specialization 050318. But on the other hand, after talking to the candidate for the degree, I can say that the author has introduced some new things and new details into the traditional models to reflect this particular data set. Therefore, I believe that the thesis of Anton Bankevich, Mathematical Models, Algorithms and Software for Genome Assembly from Complex Sequencing Data, comply with the major requirements set forth in Order 6821/1 as of September the 1st, 2016, on the granting of academic degrees at St. Petersburg State University, and Anton Bankevich can be awarded the degree of the candidate of science. The specialization is 050318. Thank you. Thank you for your comments. Mathematical modeling in biology is a challenging area, and the objects that we deal with are very difficult to represent in an accurate and specific way. Sometimes the simplicity of the mathematical model guarantees effective solution of the problems that we deal with. Therefore, I believe that the modification of the basic model will solve the tasks that we have. As for the question about software complexes, I can say that there is a method of storage in the thesis. This tool allows us to store and build the graph ruins, and this is a new and effective method. This is one of my contributions in the area. I would like to add that this is the contribution made to the biological information. It is a common method and a common approach in the database. I don't quite agree. The storage of chimeras is very important, as well as the false edges, and taking account of these aspects allowed us to develop the model. Yes, you're right, but it cannot be relevant for the software tools. Thank you for your reply. Maybe I'm not the specialist in the area, therefore I might misinterpret your achievement and your contribution. Now I would like to read out the report of the supervisor. First of all, I would like to say that when I took the thesis by Anton Bankevich, I understood that everything had to be changed. We did not take into account all the changes, and this can be seen from the critical remarks. But in my report, I focus on the following things. The algorithms work, and they are highly effective, and this is the major merit of the work. Moreover, this is proved by other scientists. I have always been interested in biology, and I have cooperated with biologists a lot, and it is always difficult to find common language between biologists and mathematicians. So in this thesis we have achieved a result and made a great contribution to this dialogue. As for the drawbacks, some parts of the algorithms have a heuristic character.
Another question arises. If we create certain computational methods and software which implements them, always there is a question how to correct of the problems that we have to solve to, first of all, increase the quality of software and to introduce some improvement. So can your software introduce improvements without breaking its total structure? Let me answer right away. Yeah, that's possible to introduce improvements. Spades was developed as a modified software set that can work with different data types. As a result, it is developed so that algorithms are compatible, parameters can be changed, and the whole process flow can be altered. Another remark, or just a moment I'd like to focus your attention on. This is something that I think. This is something about formalization of problems, something that mathematicians and physicists are used to. Biology sometimes uses these methods as well. In your study, I actually liked the heuristic character that your dissertation had. I analyzed the documents you submitted, and they showed you're not a biologist, far from being a biologist. And my personal experience shows that it's difficult to work with biological data. So rather, this is an advantage that you decided to tackle a biological issue. So our study comprises three fields: biological aspect, mathematical modeling, and software. To conclude, considering the number of papers you have written and the report you made today, you can be conferred the academic degree. I'd like to confirm that I'm a mathematician by education, but I had to dip into biology when I was developing the algorithms and doing this research. I have a question. Now the floor can be given to the academic supervisor. He's not present here, so I will read it out. It's pretty short, so I'll read it out. Amidst the introduction, what the candidate has achieved, what was done, let's omit that. We are fully aware of these details. I'll focus on the major results. They were... We have already talked about the Scopus indexed and Web of Science indexed journals. The results were reported at different international conferences, which were attended by experts from different fields. The dissertation has practical value, so the dissertation mathematical models, algorithms, and software for genome assembly from polymerase sequencing data is submitted in conformity with all the relevant requirements, and the candidate can be awarded the degree of the candidate of science in physics and mathematics. The report is signed appropriately by the academic supervisor. Now that's time for discussion. Those attending the session are welcome with their questions to the candidate. They're welcome with their comments, if any. I do not see anyone willing to speak. I'd like to make a comment. Could you please switch on the microphone? Thank you. Unfortunately, formal questions are often odd. Well, not actually odd. They are justified, yet they sound about something starts here and finishes here. There can't be any strict boundaries. I'm an expert in my field, and very often I face a challenge when what people do within a specialization is much broader than the specialization describes on paper. It doesn't matter which specialization you defend your dissertation in. I actually do not remember mine. It's just one side of it's just one side. The other side is that if we analyze the candidates' publications, then we shall see that there are not so many dissertations in this specialization. Have 6 publications with an impact factor of 2.5 and higher, up to 12 actually. 23. I haven't seen many candidate dissertation, I'm not talking about doctoral degrees, can have this high impact factor. And actually that's a privilege for us to confirm the academic degree, even if it's somewhat beyond the scope of the specialization. This is what I wanted to say. Thank you. Thank you. We are moving on with our agenda. It's time for a discussion with the colleagues. Please switch off the broadcast.
Отключите. Нет, они же должны нас слушать, они же участвуют.
Please switch off the broadcast.
Передачу. Ой. Это уже на тебе.
As a result of voting, we have the following results for 5 members of the Dissertation Board. So, sorry, this has to be an open voting. We have to also ask our members attending the session via Skype to vote openly. What's your opinion? I vote for. I also vote for. I vote for. I vote for. I vote for as well. As a result, we have 5 members who are for confirming the academic degree. So we are confirming the academic degree to Anton Bankevich. He is awarded the degree of the candidate of science in physics and mathematics. We are finishing the defense. The floor is given to the candidate of science. I'd like to thank everyone present here, members of the dissertation board, because they actually read my thesis and found it interesting. I'd like to thank my team from the lab who have been supporting me all these years, and my academic supervisor, Arkady Pavel-Persner. I would like to thank Yana Safonova and Sergei Nurka. Without them, this study would not be possible. Thank you.